talk today about all of that. Hi, Gio. How are you?
Hi, good morning. I'm fine. How are you doing?
I'm good.
Good morning, you all. My name is Gipoliano Chavez. I have a PhD in biomolecular engineering and bioinformatics. Uh, right now, I am in the University of Chicago, where I am a postdoctoral scholar. But I pursued my PhD at the University of California, Santa Cruz. I don't know if you know, but it was the University of California, Santa Cruz that helped a lot on sequencing the first human genome that was released in 2001. Before coming to the United States to pursue my PhD education, I am originally from Brazil. I studied biochemistry while in Brazil. And yeah, I am really excited to be here. I am really excited with the idea and the concept of sharing experience with people. As I will tell you later, I have been working for a couple of years as a mentor, and I think this is a great experience. Thank you.
Thank you, Gio. And I'd like to ask anyone else if they would want to introduce themselves.
Yeah, hi. My name is Dr. Taruna Anand, and I have more than 15 years of experience in clinical research, which is basically from phase one clinical trial till phase post-marketing clinical trial. So basically in clinical trial data analysis. Recently, I have started working on genomics data and also, looking into precision medicine. So I look forward to this webinar, that you are conducting, and I'm happy, to attend this. Thank you.
That's great, Dr. Taruna. It's really great to have you. And thank you for telling us about your background and your experience. That adds a lot, and actually it makes our webinar more relevant. Thank you. So we are just 1 minute away from the start of the session. So thank you all again.
Good evening, everyone. I'm Sonalika Ray. I'm the community manager, and we would be starting in a while. Thank you all for joining. Welcoming you all to today's webinar on bioinformatics for precision medicine. So let's talk about precision medicine first, and then I would want to introduce ourselves because I see many of new participants today here with us. Precision medicine is changing the way we understand and diagnose and treat as well the major life-threatening diseases. And the transformation is driven by high-throughput molecular data that is being collected from patients, animal models, and large-scale cell line experiments. In this program, we will explore how the various omics data types generated in these studies can be analyzed and integrated to study the basic biology associated with the viruses and the hosts, cancer onsets and development, and the outcomes as well. As I already introduced myself, I'm Sonalika Ray, and I'm the Omics Logic Community Manager, and I'm responsible for daily interaction with thousands of users that are a part of a bioinformatics community managed by Pine Biotech. So before we start, the talk, I would want to introduce us. We are a US-based bioinformatics company who is working with multiple academic and commercial collaborators to develop easy-to-use analytical tools. And our mission is to make bioinformatics more accessible. Omics Logic research-based training programs are a result of close collaboration between Pine Biotech and numerous academic institutions that have participated in content evaluation, curriculum review, and project design as well. Omics Logic is an international program that is running in 5 different regions with over 10,000 users around the world. And due to this fast growth, our team is working with local and regional coordinators that are helping refine local program logistics and also leverage our online training resources, adapting them to the needs of students and the researchers around the world. This online program includes weekly sessions with an instructor, online tracking and review of student progress, access to all the resources and materials online for self-paced learning, access to expert guidance and problem solving, educational and research pipelines, and also the domain data analysis on our big data server for 15 days. Let me introduce you to the program mentors. Dr. Harpreet has an expertise in the application of statistical machine learning techniques on biomedical data and the development of prediction tools and databases. And she looks after the curriculum development, bioinformatics, and data science course development. Dr. Mohit Mazumdar, as he has already introduced himself, he is an experienced bioinformatician with multiple publications in peer-reviewed journals and collaborations in industry and academia. Over the past several years, he has been developing collaborations with universities as a mentor and a project lead for Omics Logic Training and Research Fellowship. We have Dr. Cipollino Chaves with us. He's a postdoctoral fellow at University of Chicago, and he is skilled in bioinformatics and is an expert in glioblastoma. Now, I would want to pass it on to Ilya Brodsky, CEO of Pine Biotech, to talk more about the program. Over to you, Ilya.
Thank you very much, Domenica, for the introduction, and welcome everyone. It's excited to do this together today. And what I wanted to talk about is a little introduction to the precision medicine topic in general, why bioinformatics and precision medicine, but also to tell you briefly about what the program is going to help you with when you are joining this training program. So before I go there, our background is we're a technology transfer company, and we have a close collaboration with University of Haifa, the Tauber Bioinformatics Research Center, which is a center dedicated to the development of computational tools and algorithms, and also making them available in a easy to use way for many users that do not have an engineering and a bioinformatics background. And essentially the objective of the Omics Logic program that we developed is to help with training in bioinformatics, to enable independent research that is guided by mentors and peer example. We want to develop a community that is thriving and growing, and they share a passion for data-driven research and an appreciation of citizen science, which means that not just scientists doing science, but people that are learning about science are also engaged in that process. And then we also want to make sure that we are enabling students, clinicians, and faculty of all backgrounds to develop novel independent research using latest technology in life and data sciences, which means to make it more accessible and more user-friendly. And so today we're speaking about precision medicine, and I want to briefly talk about why bioinformatics and precision medicine. I mean, if you're interested in precision medicine, maybe you're a clinician, maybe you're a student in biotechnology, maybe you're just interested in applications of data for the biomedical research. And so the question is, well, why bioinformatics? So what we want to talk about today is how essentially the availability of data and high throughput computational resources and computational resources that can analyze and integrate and visualize this data is actually driving a new approach to using data for clinical purposes. And so to describe what that really means, I want to take a couple of examples, and then I'll pass it on to some of our mentors to speak more in depth about their particular examples with research studies they've been involved in. So let's take an example with cancer, right? We know that cancer incidence and mortality is growing. Specifically, it is growing in countries that have already good healthcare systems. They have a lot of research. And the major driving force behind that is the growing and aging population. So the percentage of people that are older, typically that is seen as one of the driving reasons why the number of cancer patients and mortality rates are increasing. Now, how do you leverage the ability of these countries to understand and study these diseases to address such a pressing challenge that is only bound to continue and grow and become more complex in the future? Well, one of the things that is now obvious about cancer is that the earlier we understand that cancer risk is increasing, and the earlier we can detect the specific type of cancer that is developing, we can develop markers of cancer risk, and we start addressing that cancer earlier on. And that is a proven way to address cancer mortality and treatment in a more efficacious way. And so this early diagnosis really starts very early, and maybe for some cancers, including cancers that children have, this is a very critical point. So we need the ability to screen, but obviously we cannot screen for everything. We need to know what we're looking for. And so the basis of this data-driven and biomarker-driven medicine is really having access to data. And so in the past couple of decades, I think the major exciting thing that has been happening is that next generation sequencing as a technology has been refined to the point that it is becoming accessible enough and affordable enough. So it is really entering the clinical domain. And so it is entering with this diversity and challenge that a technology like next generation sequencing can bring, because essentially next generation sequencing can be used to generate a diverse set of data. And so that brings us to this point of omics data. So why omics data? And as you know, we're called omics logic. So a lot of this program is going to be focused on omics data. So omics means a collection of the totality of something. And so it could really refer to a number of different things. For example, we can say phenomics, which is the collection of phenotypes, because really when we look at cells, let's say coming from a tumor or the microenvironment, they have different morphology. They have different characteristics that we can define in different ways. And so we can collectively call that phenomics, the collection of phenotypes about a particular organism, cell, or tissue. We can also generate genomic data, right? So a lot of us know about genomic data, genomic medicine, essentially looking for those mutations or finding associations between mutations and specific types of cancer or specific stage of cancer or specific treatment that might be effective or not. So that's called genomics. We're looking at mutations, changes in the DNA. Then there's epigenetics or epigenomics, a rapidly growing field, because more and more we're understanding that genomic regulation, what's written in the DNA, is also regulated by additional elements like histone modification, like DNA methylation, how chromatin is open or closed and how accessible it is, and many other factors as well. So a growing area of research that definitely has an effect on cancer as well, because we know that cancer is driven by genetics, but also by the environment. And then transcriptomics, where we look at gene expression, alternative splicing, gene fusion, and many other interactions between mRNA and the mechanisms that are a part of the translation mechanism that turns the written genes in the DNA into actual proteins that are being used. And not only is that a useful proxy to look at proteins, but an important view of what are the specific biological processes that are happening right now in a given group of cells, individual cells, tissues, organs, or the full organism. And so what is, I think, exciting about availability of this data is that it provides us with a very detailed view of not only the result, the product of what is in the DNA, but it actually gives us a snapshot of what is being done right now. So if you think about how this affects clinical use, you can imagine that if before we were looking at expressed proteins, if we were looking at histology, if we were looking at morphology of the tissue and differences in the phenotype, now we can look deeper inside every single cell, and we can actually understand what is going on there right now. So how is this changing cancer treatment? Well, one important factor is that it is actually right now changing breast cancer classification as one example. So not only are we looking at the changing practices in the clinic because of next generation sequencing data, but we're actually looking at an active area of research that makes a lot of these clinical approaches to treatment, diagnosis, and prevention of cancer a dynamic area of research. And so we're really looking at now how we can look at histological classification, functional classification, and molecular classification together by integrating multiple types of omics data through omics integration, but also then how to use machine learning on large data sets that we already have available to integrate and make sense of this data in the context of disease classification, but also for individual patients. And I think that's why really we're looking at an important and a growing role of bioinformatics and informatics and data science in general in this clinical field. So it's really about bridging the early stage basic research using this data to translate this research to practice and understand how different aspects of omics data could be applied in practical clinical situations. Now, that's one level of complexity, multiple omics, multiple types of challenges. But another one is that really we need to connect how this basic type of research really connects to what we see in patients. And typically that means how do we translate insights from patient-derived cell lines to studies that are being performed on animal models, to then the application of that information for clinical trials and also into the clinic, which is the ultimate goal of genomic medicine. So a good example of that and how it is changing not only with omics data, but with omics data as well, is this application. And here's an example where we are looking at existing types of cancers. We're looking at new types of disease states that we can identify. And ultimately, instead of approaching that from a typical clinical perspective, we're really looking at a data-driven perspective where we are trying to actually leverage the availability of data. And a lot of that data is available in many different repositories, and I'll briefly mention that, to essentially speed up the new development of treatments and clinical applications to leverage that data, treat patients better, develop new or repurpose drugs to treat disease better, and also to even prevent data by understanding better what are the driving risks behind many of these types of diseases. So as I mentioned, surprisingly, not only is this data available, the availability of data is growing at a rapid speed. And so what we see, especially in this pandemic, right, we've seen how the availability of genomic data, not only is it growing in volume, it is also growing in how fast it is becoming available, right? So if you imagine as somebody who is maybe a student or a clinician, you know, where do I get all of this data, right? So there's so much data becoming available every day that we can mine and learn from and understand. It is amazing that we can leverage such expertise and investment of others into generating high quality data sets, really generated from, like I mentioned, cell lines, animal models, as well as patients. But the problem is really that we need to learn how to navigate this data, right? So here in this image, you can see this is mapping of reads to the reference genome, right? Every single box right here represents a piece of data. So working with this data and making it useful, really extracting some meaningful insights is challenging. And that is why we can hear about all of these exciting methods, people, doing amazing projects and maybe integrating a lot of this data and running all kinds of artificial intelligence on it. And the problem is that it seems out of reach, right? It is just complex because you have to deal with huge amounts of data and very complicated methods. So how do you get trained? How do you get into something like that? Or if you're already in it, how do you expand your horizon so that you can actually leverage some of these more advanced methods and speed up the process of utilizing bioinformatics to actually ask such questions and do some kind of meaningful work in this field? And so our solution to this is to try and make tools for analysis and integration of this data more user-friendly, and to train people with the logic behind how these tools work so that anyone can play around with big data sets, analyzing and integrating them together, asking research questions that make sense and that will essentially help you understand both the data and the computational approaches in a better way. And so what I mean by that is that we built the logic of analysis into the toolkit that we have, and we'll talk briefly about some of the resources that you'll have access to in this program, where the logic is explained in courses and applied in a meaningful way in a computational big data analysis platform that you can point and click, getting quickly from raw data sets to structured data sets, developing tests and annotating them with biological information so that you can quickly go from raw data available on these public repositories to biologically meaningful results. And so this kind of annotation, I think, provides us with an opportunity to make complicated methods that ultimately are required to make sense of this data easily approachable, and we can now take examples from real studies that will allow us to ask these questions and understand what others have done. For example, availability of the Cancer Genome Atlas, or the TCGA, really is one of those examples where we can source multi-omics data from 12 different tumor types with thousands of patients that have information about their clinical stage, about the different types of tumors, about the different types of treatments that they received, and even comorbidities in many cases, and start asking meaningful questions about how that data could be used to inform the classification of patients, the treatment strategy, the prevention, the risk, right? So all of these important questions could be asked. Not only that, we will also take a look at some examples where cancer studies using patient-derived cell lines could be used to train mathematical models using machine learning and classification to start predicting different classes based on signatures or specific molecular biomarkers. We will also dive deeper into some of the clinical data for risk factors and outcomes. We'll take a look at publications in top journals like Cell and Nature Medicine that actually show us how do people utilize some of these types of data to come up with new computational methods, but also how to apply known bioinformatics approaches that you will learn in this program to extract meaningful insights. So cancer obviously is one of those examples. Another example, a type of diseases that we know is infectious disease, is also a growing area where molecular precision is becoming more and more informative. And I'll give you just a quick example. We all are hearing about these new variants coming up in different strains of coronavirus all the time. And what we know now is that these strains are defined by mutations, and those mutations have a critical impact on the rollout of vaccines, right? We actually hear about that every day in the news. And it's amazing that today genomic data is so easy to talk about, even though just 5 years ago, you would start talking about genomic mutations and people would just stop listening to you, right? So we live in an era of genomic data becoming more and more relevant to everything that we interact with. And so this application of molecular biomarkers and genomic variants to insights that have a practical public health and also clinical impact and significance is now very obvious. So this program is going to talk about some applications of computational methods to a variety of clinical, biomedical research, pharma and biotechnology challenges using big data that is sourced from peer-reviewed journals and found in different repositories that anyone can get data from. And it is going to be oriented towards where this field is going. And it's obvious that this field is going towards big data, where data mining and classification methods are becoming more and more user-friendly and easy to apply to these data sets. Extracting meaningful signal from that data is becoming easier. But at the same time, what you really need is you need the logic of bridging the gap between the biomedical and the clinical applications of this data to the actual methodology so that the two can be used together in a meaningful way. And so in this program, we're going to go through a lot of these examples with some also technical, practical training that you can get access to, how to perform many of these analytical approaches on our big data analysis platform, including in R and Python, learning from curated examples, and then developing a sense of a computational process where you go from raw data all the way to biologically meaningful insight, and taking that with you so that you can start developing an environment for yourself where you can easily leverage this data for meaningful biological and biomedical research. And so some of the outcomes that we envision for those that are joining this program is to understand scientific literature that describes analysis of omics data, to know how to do basic processing steps for next generation sequencing data analysis with consistent accuracy and meaningful approaches, to perform exploratory analysis and generate a hypothesis from data, to design contrast for statistical tests and prepare data for machine learning, annotate found signal using biological datasets and references, identify appropriate datasets for answering a biological question, how to plan a bioinformatics analysis from start to finish in an independent way, and then how to present a project proposal, including the rationale, the methods, scientific background, and anticipated results. And so this, if this is relevant for you, and I want to mention that we've previously had hundreds of people finish this program and really taken away meaningful training skills that they could later apply to a variety of challenges that they face in their own research. And so that includes many different participants of different backgrounds, including high school students that are ambitious and want to get into a program with an advanced project that they can present. Includes undergraduates that are learning something relevant and really want to get ahead and maybe want to prepare for their master's studies, or they want to go into a more data-driven field. That includes PhD students and postdocs that are now engaged in different types of data analysis projects, and they need the skill set and the tools and resources to make their understanding of that project more data-driven and apply these tools in a meaningful way. And of course, researchers and faculty that are also trying to leverage bioinformatics to make their research more impactful. So now I would like to pass it on to Cipollino Chavez. He's going to be one of our mentors in this program that you will be able to interact with. And if you are interested in developing a research project, work with him on that part of it, but also hear from his experience in research. And I know that, Cipollino, you come not from a bioinformatics background. You actually learned bioinformatics as you were getting interested in your research topic. So I'm excited to have you here, and maybe you could share a little bit more about your research interests as well. Thank you, everyone.
Thank you. Thank you, Elia, for this very nice introduction. I'm going to share my screen now. You all see my screen, correct? Okay.
Yes, we can see it. Thank you.
I think I said your name incorrectly. It's Elia. I'm sorry.
No problem.
So good morning, everybody. My name is Cipollino. Currently, I am at the University of Chicago, and I'm going to talk to you a little bit today about the challenges that I face on my research, and really based on what Elias said, that I do not come from a bioinformatics background. I want to give you an idea of how you can get introduced to that and how you can get started with that. So to start, I'd like to tell you a little bit about my mentoring experience. I have been a mentor at this point in the summer internship program at the University of California, Santa Cruz, where I got my PhD from. I was a mentor there for 5 or 6 years. And in the right part here, you can see that I not only mentor high school students, the SIP public, the cohort that of students that used to take part on the summer internship program is basically formed of high school students. But it is really, really great to also mentor and have the opportunity to interact with the early educational students at UCSC. My son is actually part of this cohort here when we were touring them and showing a little bit of science, of what it is to be a science, what it looks like to be a scientist. And so it was really, really nice to have these kids interact with the lab. So with all this experience, I think it's very important to have a sense of community for the human development in any area that you work, in anything that you do. And particularly in this field that we are at, the bioinformatics field, it is also important to teach kids and adults computational concepts because it's a form of investment in their lives. As we said, I come from an undergraduate background where I didn't have any exposure to computational programming. So it's really important to provide people with that type of training in a field of science that in my mind, and I guess many of you would agree with me, is so needed these days. So for many of us, and for me, particularly a computational algorithm is a thing that I never used it to think. 5 years ago, for instance, before that, I never used it to really think about what a computational algorithm is. But really today, I would like to give you a very general idea of what a computational algorithm is. It is basically a set of rules to be followed in calculations to obtain results from as an output from a given input. So you basically have some piece of data, you give it to a computational algorithm, and you get a result out of it. And thank you, Elia, so much, because you talked about this concept of classification, how it is so important in cancer and in these advanced genomic studies. But really, I like to start from the very simple principles of science. And I guess we have been using these some sort of classification for a really, really long time here. This example of classifying, for instance, animals. So we can, for instance, think about classifying the animals based on their number of feet. And I hope I'm saying that correctly in English. So here we have animals with 2 feet, and here we have those animals with 4 feet. And this is a system of clustering or classification. We could argue that in that regard, classification system, like classifying the animals by their species, is a more refined way of classification compared to clustering. But really, we are talking about grouping subgroups based on similarities that they have. So how can we get started with this computational classification, I would say, that we are so interested in? First, I would say it is really, really important to identify a problem. As I am going to tell you, the problem that I work with is cancer, particularly neuroblastoma, which is a pediatric cancer. The other thing that I would say is important is to identify a sponsor, so a mentor, a school or a group, a science group that you might be interested in. And really, what you need is someone or an institution that has the potential to guide you through the process. And frankly, I joined Omics Logic a couple of weeks ago. I'm still getting used to all the resources that they have, but so far, I think the resources that I have seen and have tested and used, they're very, very valuable resources in the bioinformatics field. So I said that it is important to identify a problem that you're interested in, and the one that I told you I am interested in is neuroblastoma. It's a complex or polygenic disease. I will not go too much into details, but I want to say that it's a problem in the development of the sympathetic nervous system. So the sympathetic nervous system is this part of the nervous system that connects organs to the central nervous system here in the spine, for instance, as you can see. The other problem that I want to go to in neuroblastoma is this concept that we have two major populations in the tumor. So we have the whole mass of the tumor. We could argue that there are many subpopulations, but I'm going to tell you about the two main subgroups, the two major populations, which are the population of adrenergic cells that look actually like neurons. That's why you see these projections from the cytoplasm of the cells, making them really look like neurons. And we have the mesenchymal cells, which do not have these projections. They are not. They do not adhere. Sorry, they adhere very tightly to substrate. That's why they have this name of S-type, which means substrate adherent. But the difference between these cells is that the adrenergic cells are really differentiated and the mesenchymal cells are not differentiated. You can also think of them as cells with a stemness potential very high. So these mesenchymal cells are not differentiated. They are not sensitive to chemotherapy. They do not die upon chemotherapy. And they lead to this concept of minimal residual disease, which is the concept that after you give chemotherapy treatment to the cells, the chemotherapy is going to kill the adrenergic cells, but not the mesenchymal. And the mesenchymal cells have the ability to differentiate into adrenergic cells. And that there we have a problem because the chemotherapy is not going to kill the cells. And so there is a chance that the disease is going to come back. And when the disease progresses or relapses, there is an enrichment of the mesenchymal cells. It is really important to classify neuroblastoma tumors, and I'm going to give you a more detailed idea of what that looks like. It's really important to classify the tumors into adrenergic or mesenchymal because that has a potential for prognosis. But so far, I mean, that's the part of research that I do. Here, this slide shows you what really the ultimate level of prognosis is in neuroblastoma. So a neuroblastoma tumor is really classified into high risk and non-high risk neuroblastoma. So the high risk has a very lower survival probability compared to non-high risk. And really one important problem is that after the tumor of a child is classified as a high risk neuroblastoma, there are no methods to determine whether that disease will progress or not. So measuring, as I said, the mesenchymal cells may provide prognostic ability to the research. And mesenchymal cell quantification can be done by sequencing of epigenetic profiles. So clustering consists on the identification of homogeneous groups among a set of heterogeneous items. In the specific case of neuroblastoma, what I do is I classify cell lines or tumors based on their gene expression profiles. The other thing that I have worked with is classification of SARS-CoV-2 based on their mutational profiles. You would agree with me that mutations are important in cancer and are also important in COVID-19. So for neuroblastoma, this is an example of what's going on right now in the field, because what people try to do is to classify these adrenergic cells and the mesenchymal cells. This is a machine learning method called hierarchical clustering. So we can basically separate, classify, or clusterize the adrenergic and mesenchymal cells. We can do the same using what are called wet lab methods, such as a Western blot. This is a biochemical method of protein quantification. And you can use some scoring systems to classify adrenergic and mesenchymal cells based also on their gene expression profiles. This image here in the right is a similar method. This is also a hierarchical clustering method, but this is based on enhancer peaks or cis-regulatory regions, the enhancer regions that we use to classify or differentiate the adrenergic and the mesenchymal cells. So what I do currently is I basically score tumors. So what you see here in the left are 498 tumors classified with a high mesenchymal score, top high, second highest mesenchymal score, third highest mesenchymal score, and fourth highest mesenchymal score, which is basically the lowest mesenchymal scores. And what we can demonstrate or see is that tumors with high mesenchymal scores, they have a lower survival probability. Here I'm looking at all risks, but there is some evidence that when I look at the high risk cohort, that cohort also has a lower survival probability. And so this is showing some evidence that the classification of tumors based on their epigenetic profiles may allow some prognosis analysis in high-risk neuroblastoma. And this is very important because, as I said, for high-risk neuroblastoma, there's no such a prognosis system yet. And that's what we're working with. So I am going to end now my talk by showing you one thing that is really interesting in my opinion, which is the fact that we can compare these very different types of diseases. On the right, I have a heat map of neuroblastoma. This is using the RNA-seq profile of neuroblastoma. Sorry if I said this is the left, this is the right image. In the left side here, we have a heat map showing the mutational profile of COVID-19. So with the high school students that I told you, we developed this classification system of COVID-19 based on the mutational profiles. So here what's interesting to note is that by looking at the frequency of these mutations, we see some sort of clusterization of the mutations that are in the extreme east of the Earth, such as the samples collected from China, also present in the bat and the pangolin, which are thought to be the animals that are more permanent hosts of the virus. So really here, what I want to say is that computational biology methods allow us to compare or to have a sense of common analysis that can be done in two completely different diseases, such as neuroblastoma and COVID-19. So in conclusion, as I said, bioinformatics can be used to study and understand these two very different diseases and many other types of different diseases as we saw during this presentation. And more specifically, in my case, bioinformatics may allow the development of prognosis system for high-risk neuroblastoma. So I would like to end now. Thank you so much for listening and for the opportunity to talk here.
Thank you so much, Dr. Cipollino, for thank you. Thank you so much, Dr. Cipollino, for sharing your research work. It really sounds very interesting. Now, I would want to welcome Dr. Harpreet, our mentor for the program, to please introduce herself. Over to you, Dr. Harpreet.
Snalika, can you give me some time? Actually, my system is a bit hanged.
Yes, ma'am, sure. No issues at all. Now I would want to ask Dr. Mohit Mazumdar to please share the session details.
Okay. Thank you, Snalika, and hello, everyone. So before I share the session details, I'd like to thank Ilya for this excellent overview, and it helped us learn a lot of new things. and Gio also for the introduction to the research. Uh, now, I'd like to share my screen to show you some of the resources that are associated with the program. And it's just for everyone, I think, because we do hear about some of the confusion that are there with all the resources that have been shared. So I just wanted all of us to go through some of these resources and to understand where to find the things that we discussed today. So this is the pre-registration page where we all started, and like many of you have pre-registered here. So in this page, we have a video overview, and then we have the session details. Along with that, there's a form that many of you have filled up, and then we send out the emails. So when you go through this page and you would want to learn more about the sessions, so the second thing that I would like to take you to is the program syllabus and how we have planned the live sessions and the resources that are associated with it. So one of the exciting part of this program is just which also differentiates us from other similar online programs, is that there are several resources that are associated with each of the programs. So this is the syllabus page, and Sonalika, if you can paste the link of this page too, so that we can all browse through this page. So as we go to this page actually describes the curriculum that we have developed, and also the resources that would be associated, including the assignments, the hands-on sessions, and what will be covered in the hands-on sessions, and where would you learn about those technologies, and what all Ilya just said about getting started with big data, getting started with gene expression data, getting started with um, genomic data, and then applying it for cancer problem and applying it for infectious diseases. So there's a lot to learn, and there is, so I mean, I'm really happy that we have all of you together today to showcase some of these that we have put together for this program. So there's a video again which gives an overview of what was covered. So you have already seen what was covered. So session wise, when we go to when we will start, which would be on Tuesday. So we are going to start next Tuesday at 9:30 a.m. CST. I think it's 10 a.m. CST. So yeah. So in the first session, we will be covering about bioinformatics and translational oncology research. So this would be the overview of the cancer problem. As we were discussing, we will discuss about the complexity in the sense of how it is being described at tissue, organ, and cellular level, and then the molecular factors that actually are associated with tumor development, growth, and metastasis. Then we will understand that the how genotypic and phenotypic relationship can be identified through these clinical and molecular data types. And then we will go through the overview of the platform and what we will be doing throughout the course. So this will be on the first session, which will also be available to all of you. And the associated online resources, as I was telling you, that we started off with the learn site. So let me go to the learn site. This is the associated courses that are available on the learn site. So if you have not created a login for yourself, so please do that. This is the link, and let me show you how you can do that. It's very simple, and this is also also for those who would not who are participants, who would be participants, and also for those who would think about joining later. But it's great to have you here. It's a free login, so you can just log in with your email address or your social media address. So I'll just sign in. So once I sign in, I'll come to the home page. I'll just show you how to navigate through this website. So once you come to the home page, it describes all of what we described and discussed today, as well as what are different specialization that you can opt for. So we have specializations in oncology, infectious disease, neuroscience, agriculture, and then the T-Bioinfo server, and also all of the things that we discussed, our associations. And then the interesting thing about the syllabus is that we will start with introduction to bioinformatics. So let's go to our first course, introduction to bioinformatics, which will be covered in the first session. So in the first session, we would want to learn, we would all want to learn about where bioinformatics can be applied, especially in biomedical research. So all the areas that you might be interested in, you will be able to navigate through this. And this course also has a form which let us know more about you, about your research interests. So this is a course that I recommend you all to take, and it's a free course. So once you create your login here, your free login here, you can start taking this course after your login is done and you have a profile. So let me quickly show you what a profile looks like. So this is my academic profile. I have a image, and I've described myself. So one of the benefits of having a profile is that you will be able to see your progress, how many courses you have finished, how many programs that you are enrolled in, and also like what are the, as I said, that what are the courses that I'm enrolled in, I'm doing, and what are the programs I'm enrolled in and I'm like interested in going through, and then the certifications that I've gained from taking the courses, these courses, or completing the programs. So all of this you will gain and get from this profile once you just create a free profile here and put some of the information that would help us learn more about you, and also also people who are interested to collaborate with you, or people who are interested to hire you for any specific research work. So all of that is actually are the components of having the profile. So this I would really recommend you to all of you to create and at least go through the free resources that are available with all of the sessions. So as I said, that the first session that would be about introduction to bioinformatics and introduction to cancer. The second session is more technical, where we dive into genomics. So in genomics, we will learn about next generation sequencing. We will learn about how we can analyze genomic data and find out those variants and those variations and those mutations, significant mutations. So again, there is a bunch of courses that explains genomics, and one of the benefits of doing this is not just that you would be able to learn from the very beginning, from the very basic, starting from the DNA structure, but also you will have an option to work and learn R and apply R or Python using our coding playground tutorials. So here I will show you another demonstration where you will able to also apply data science techniques. So about genomics, you will be also be able to go through the hands-on sessions, and the hands-on sessions with the star mark are the ones that are available to the participants, and also to those who would want to subscribe for the asynchronous material. So when I click into this, I'll get information about how I can prepare my pipeline and understand about the process of creating a pipeline and what are the components that are involved. So once you complete, this is like an achievement that I've gone through this tutorial, and I have gone through this tutorial. So, but not yet on the platform. So here you will also learn about the data sets that are embedded in the with the link that you will get. So these are all the benefits of getting to the asynchronous material and going through the tutorials, and that goes from basic introduction to building up variant pipelines, to do a phylogenetic analysis, and again go through multiple sequence alignment to see the variation and evolution of genomes. So obviously there are so many applications which I cannot list out in this session. However, the second session and the third session after the second session learning about genomics would be about transcriptomics data, so gene expression data. Here also we will go through an overview of how what is the significance, what is RNA sequencing, why it is done, its history, and then about its application, that how do you do it, and like get understanding about the quality of the file that are available, and then how do you pre-process the data, map it to the genome, and going through all that process. So we will be doing this on the T-Bioinfo platform. So just let me quickly go to the T-Bioinfo platform. So sorry about this. This is a different pipeline. So this is the T-Bioinfo platform, which we discussed in this meeting, and which is a really amazing platform, which is a very comprehensive as well, describes all of the NGS data types, and shows us very elaborated methods and pipelines to analyze this sort of data. So this is a typical pipeline. This is how it looks. And one of the best part of this is that it's the entire platform is very friendly, because it describes all of the processes, all of the data types that are there, and what are the applications that we can do with this. So when we go into this section, this is about transcriptomics, and I was discussing session about the transcriptomics. So here we will be going through the example data sets that we have compiled for you and learn from. So this is a pipeline which has already inbuilt data sets which are put together, and you will be learning about those data sets in the session. And then we will be going through the pipeline together, building a pipeline, how this format makes sense, why these are placed in this manner. So the flow is that these algorithms are placed in, I know many of you are already aware of this, but for those who are not, are in a logical manner, put together in a logical manner, where we start from pre-processing the data to then error correction, then maybe mapping it to the genome, or doing isoform construction, or doing mapping it to the transcriptome, or doing differential gene expression. So there are several experiments, I mean, you can build from different paths that you can take to analyze your data. So in this session, we will be learning about the breast cancer sample cell lines that we discussed, and the publication that is associated with the session. So I think I can go about it a little bit more, but I think we don't have that much time. But coming to the part where we will be discussing about the application, and there was a question about classification and clustering in the chat. I think it is something that also I would love to talk more and more about, but we already have a session described for this, where we will be going through machine learning. And in this, we will be applying gene expression data and finding patterns from the gene expression data. So in this process, we will be applying machine learning methods using the T-Bioinfo server, obviously. So we have those very well defined in these sections of data mining. So you will be able to do clustering and PCA, and also if network. But yeah, you can start with all of these methods when you start with the program. And also, like to those who are joining, I think it's great to start with the coursework and get a glimpse of the sessions before actually the start of the program. So now I would also want to tell you more about a little bit of machine learning in biomedical data and data science. So what is the data science aspect? So sorry, let's go back to the courses. And let me search for data science. So we have a lot of data science courses, and one of the example I would want to show you is that Introduction to Data Science for R. So here we have a lot of tutorials that you will be able to go through, and it's relevant to the session. So we will be going through some of the visualization techniques. How would we do PCA and using R code? So as I said that RStudio is available here on the browser, so you can actually learn about the problem here, and then learn about the data that is available, and then the commands where it is described what it explains, like what data set that we are taking, what kind of step that we are doing. So here, once you learn about these steps and learn about what will be done with the data, you will have an opportunity to do it yourself. So here's a big tutorial. So learning about all these techniques, going through the assignments, and you'll be able to run it yourself. So this is a code block where this you can run the scripts by learning about the scripts and then actually doing it yourself and getting results. So for the data sets that maybe our researchers are working on, our research fellows are working on, they're utilizing these tools to come up with images and for presentations and publications, etc. So this was about that. And then the fifth session is about oncology, where we will be learning about the data, oncology data sources. So it's more about the data repositories. So we will be going to NCBI. We will be looking at the data sets that are available there, learning from them, taking some of them as examples and understanding how they connect. I mean, how do you annotate your results better? So how do you annotate your mutations? How do you annotate what information that we are getting out of the machine learning after the machine learning analysis? So this is the first phase. After the first phase, second phase is all about infectious disease problems. In this, we will be learning about viral genomes and host transcriptomes. So we have also virology sections that will be utilized in this program. So going back to that. So here we will have examples projects in this time. From this part, you will start working on projects. You will have an opportunity to start working on projects. So these projects are asynchronous projects that you can take anytime, and you will have an opportunity to work them through when you have availability. And these are like very descriptive projects that describes yeah, I mean, these are some of the data sets that are described by publications, and we have put together how do you analyze and understand from what the authors have described. So these are some of the example projects, and then we will learn about genomic multiple sequence alignment and phylogeny. And we have another few projects like COVID-19 and Ebola. So these are again about the mutations and how COVID-19, or I mean, this is from a paper that describes the proximal origin of SARS-CoV-2. It was published in Nature Medicine 2020. So yeah, you will learn about this, you can learn from this project as well along with the session. So these are the live sessions that we will be covering, and along with that there are always asynchronous material available for you to take throughout the week. And then we will go through project examples where we will learn how to compare and I mean, these are different biological questions that we are asking from the data, so that you get an experience of how can you ask your question from your research. So again, like after learning about all of this, the last part of the session, the 10th session, is about host pathogen interaction. So here we will learn and connect some of the aspects of our omics data to structure, and finding those key mutations and understanding those, and then the effect what it caused for the function of the protein. So this is what is planned for this precision medicine program, and I hope that I've been able to describe this well to all of you, and I'll be happy to answer any questions that you have.
And also, I wanted to add that signing up on the Learn portal, that is learn.omicsproject.com, would get you the automatic access of the Bioinfo server as well, which Dr. Mohit just shared. And yes, so we also share, I would be mailing you all the details which were discussed in this session, and also would give you the links and how you can proceed further with your submission.
Right. So thank you, Sonalika. I just wanted to add a couple of things. So we also have a Slack channel, and I forgot to mention about the program part. So yeah, when you go to learn and when you do this login, you'll be able to go to this tab programs. So in programs, we have all the programs listed out that are upcoming or have been ongoing. So you will find precision medicine here. And to register for this program with live sessions and everything, not just the asynchronous material, you have to start your application. So using this link, and once you start your application, you can select different options. So these options involve all the cloud access. So there are 5 terabyte of space that would be allocated, and you will be able to run the cloud pipelines, and then the coding sessions. All of this is included in this package. And here you will be also able to apply some of the discount codes or some of the fee waivers that have been provided by Sonalika. And also for lower income countries, we have a scholarship option that is also available. And I would like to request those who are applying for scholarship to do it as soon as possible, because we are going to start the program on Tuesday, so we can process your application by Monday. So I would also request Sonalika to share the scholarship link. So there are different access levels, so different licenses. Another license is about the intermediate step where you would want to have an additional one-on-one mentor support, where you would want to go through some of the materials along with the mentor, ask your research question. So even in this entire program, and even in the webinars that we do, it would be really nice to discuss your research question, but yeah, obviously there's a limitation of time. But when you are in one of the programs, then it would be it would give us like a lot of time for you to discuss your research and work on your idea, and if we can utilize these resources to be able to come up with the research outcome. So the research part is also being in this advanced license, so it gets you to work on your own project and additional month of cloud access. So this is the advanced license, and then obviously there you will have access to a lot of courses. And then once you go into the sessions and like register for the program, you will be able to see a different interface. So I'll take you there. But you will also be able to come up and it will also be able to get your certificate, as I showed you earlier. So this is the registration part, and along with this, now let me show you. I'm not registered for this program, so I would just want to show you another program and the interface. So once you're registered, this is the interface that you get. So you will get the courses that you're doing and all of that, and you can ask for a Q&A, or you can ask for a mentor session, and then you'll get your certificates here. And when the sessions are ongoing, you will find the session videos here. So the video recording, you will get into when you get registered, and then you will have access to an interface like this. And then you will be able to get the session recordings into this interface, so that you wouldn't have to go anywhere. It's a same place where you will find all the details. So also the same thing for the Zoom link for the next session, you will be able to find the Zoom link as well. So this is another program that is ongoing for our partners in Louisiana. So it's a program with LBRN network. So I think, yeah, that's it that I wanted to tell you all. If there are any questions, I'll be happy to answer those. And now I would also like to invite Dr. Harpreet to tell more about herself.
Thank you, Dr. Mohit. Let me share my screen.
Answering your question, Vipra, yes, the sessions would be recorded and they would be given the access to all the program participants. Yes, Dr. Harpreet, you may continue. Thank you.
Thank you, Sonalika. So, hello everyone. My name is Harpreet Kaur. I have a PhD in Bioinformatics. So, my PhD was in the area of cancer genomics, particularly liver cancer. So, my PhD, sorry. So, the major areas that I have worked on during my PhD, that was uh, biomarker discovery for the diagnosis and the prognosis of the liver cancer. Here, I have mainly focused on accurate cancer prediction, particularly first hepatocellular carcinoma, which is primary liver malignancy, and the second cholangiocarcinoma, which is another primary liver malignancy. Then, in the another area where I have worked, that is, I have worked to identify important genomics and epigenomics biomarker for the early prediction of SCC patients. In the next project, I have worked on the area to stratify SCC patients based on their reoccurrence, that is whether there is a low reoccurrence risk or high reoccurrence risk. So in my current role, I'm in my current role, I'm curriculum developer, and so I'm involved in designing research project and assisting the participants in their various types of analysis. I'm also mentoring number of the research fellow in their research projects. So I'm looking forward to work with you during this upcoming program. So now over to you, Soralika.
Thank you so much, Dr. Harpreet. Thank you so much, Dr. Harpreet. And thank you everyone for joining today. And if you have any questions, you may mail me at marketing@pine.bio. Or you can also mail Dr. Mohit at mohit@pine.bio. And we have shared all the details with you. I would be sharing the session recording as well. The webinar we had today, the recording of the webinar, and also all the details, including the program registration page and the pre-registration page as well. And I've shared the mails. If you have any doubts, you can ask us. Thank you so much, everyone, for joining.